Hello, everyone. Anybody in the audience? We are live with our Journal Club 9. And today we have a special guest with us, Dr. Juan Inclan-Rico, who is a postdoctoral researcher right now at University of Pennsylvania. And he has some very cool findings to show us about type 2 immunity, especially innate lymphoid cells and basophils. So at this point, without waiting anymore, I'll transfer the control to you, Juan.
Sure. Yeah, good morning, everyone. I'm very happy to be here. Thanks for the invitation. I think you have to give me permissions to share my
Yeah,
screen.
it's done now.
Oh, cool, cool. Okay. Let's see, share. All right. Okay, good. Yeah. So as I was saying, thank you very much for the invitation. I'm happy to be here and show this data. Just wanna make sure. So as it was mentioned, I am a postdoc in UPenn now, but the work I'm going to present today is part of my PhD project that I did in Rutgers in New Jersey Medical School. And at that time, I worked at the laboratory of Mark Syracuse, and we really study helminth immunity. So before I actually get into the big paper, I wanted to take this opportunity to speak a little bit about helminths, right? Helminths are not, I would argue, the usual pathogen that it's covered in your immunology classes. These are large multicellular parasites, right? They truly come in all shapes and sizes, as you can see in these pictures here. And despite this diversity in their morphologies, they also have really diverse and complicated life cycles, right? Involving multiple hosts, involving multiple organs in their mammalian host, different larval stages. So they are quite complicated. And one of the messages I try to convey when I speak about helminths is that these pathogens, they require the host, right? So, I mean, in order to continue with their development. So it's really important to maintain a balance between the host immunity um, and the success, I guess, of the parasite. Um, so these pathogens are actually quite distributed around the globe. They affect approximately 1/4 of the global population, especially in tropical areas of the world, as you can see in this diagram. And on top of that, elevated infection rate. Most of the time, these infections I mean, there are therapies to treat them, but they often result in reinfections, and there are no vaccines to treat these diseases. That is why there is an urgent need to develop these new therapies to combat these diseases. And in the Syracuse lab, we believe that understanding the immunity against these pathogens can inform that. And what's interesting too is that despite the diversity that I just mentioned of these pathogens, the immune response against these organisms is very dependent on types of cytokines. So what I mean when I talk about, or what we know about this type of cytokine response, I know this diagram looks complicated, but let me walk you through it. What we know is that after helminths colonize mucosal epithelium or mucosal sites, as you can see in this picture here, such as the lungs or the gut, they induce vast amounts of tissue damage, right? Remember, these are large organisms. They induce a lot of tissue damage. And they also secrete a number of helminth-derived molecules, and it's well established that many of these are focused to modulate the host immune response. Again, reinforcing the idea that these type of organisms, they require the host for their success. So what we understand about the type 2 response, is that this tissue damage induces the activation of different innate immune cells, such as the group 2 innate lymphoid cells, and I'm going to go into those in a minute, as well as dendritic cells. On one hand, the ILC2, in response to these signals, they secrete robust amounts of these types of cytokines, namely IL4, IL5, IL9, and IL13. On the other hand, dendritic cells that are activated by these helminths, they promote the polarization of naive T cells into a Th2 phenotype. In these type 2 cytokine microenvironment. And then later on, the Th2s further contribute to the production of these type 2 cytokines right here. So what do, right? So these cytokines induce a number of effector mechanisms, including the activation and recruitment of other effector immune cells, such as eosinophils and mast cells, as well as the production of mucus by goblet cells or the polarization of macrophages into an M2 phenotype. And some of these readouts, especially the eosinophil and the mucus production, I want you guys to keep in mind because we're gonna go back to those in the paper. But importantly, these effects, right, these cellular mechanisms, they have two main functions. One of them is to promote expulsion of the parasite out of the tissues, right? The other one is to actually repair the damage, right, that I just mentioned that these pathogens induce in those mucosal sacs. And these two processes, they are dependent on the type 2 cytokine response, and that has been established before. So a lot of focus has been put in the recent years in the field to understand how these mechanisms initiate, right? How this type of cytokine response needs to be initiated in order to promote resistance against the parasites and control the worm burden, right? However, equally as important, I would argue, is to understand how these responses are limited, how these cytokine response doesn't go crazy, doesn't go overwhelmed. And the importance of that is because when you have too much of that helping induce inflammation, this can result in immunopathologies, specifically in the case of helminth infections and type 2 cytokine responses that can be illustrated by the fibrosis that develops in helminth infected patients. So it's really important for the host to promote an immune response that is strong enough to control the parasite burden, but limited enough in order to do it without overwhelming the system, right? To put a brake on it.
Right.
And that was,
Oh no, just agreeing with it.
okay. So that was really where we were when I started my PhD in Mark's lab, right? How are these responses properly regulated, right? And please stop me if you guys have any questions. So with that in mind, really my project started with at the time with a very specific question. What do basophils do? How do they contribute to the proper regulation of this cytokine response, right? And we were interested in basophils because, and this is one of the reasons why I like this illustration here, was because for a long time we've known that basophils, this population of cells expand in numbers in the context of type 2 cytokine responses. But what they do is not entirely clear. That's why they are, just put here in the tissue. We don't really know what they do, right?
Can I add something there?
Yeah.
Just for an educational purpose, and I work with cytokine signaling and JAK-STAT signaling specifically. So because if you look at these numbers, IL-4, IL-9, 13, they look like these are random numbers, right? It's hard for people to figure out how could they be, like, they're named in a way that's just confusing to people who are new. So something that's common about these cytokines, like 9, 5, 13, and 4, is that they all signal through, or mostly they signal to STAT6. STAT6 is downstream of Janus kinase. Yes, and STAT6 is the common factor between all these cytokines, and STAT6 itself is a transcription factor, and it also recruits these chromatin remodelers that will open accessibility in the chromatin to a whole new area of genes that will in then give you production of these very specialized functions like tissue repair and mucus production. So it if you look at from the intracellular perspective, it makes a lot more sense that these are all cytokines doing similar things. Not exactly. I don't believe they have overlapping functions, right? They do have some unique functions.
Correct. Yeah. They have some overlapping, but they also have very specific functions. For example, IL-4 is very known to induce the depolarization of macrophages and T cells into Th2 and M2 phenotype, respectively. But IL-4 is also very related to the class switching of B cells into the IgE producing plasma cells. IL-5, for example, is mostly recognized to induce eosinophil development and from the bone marrow. IL-13, it also contributes certainly to the polarization of Th2 and M2s, but it really signals quite heavily on the epithelium and induces this production of mucus by goblet cells, the contraction of the smooth muscle cells. What's actually interesting about 4 and 13 is that they share, it's a heterodimer, right? It's two, but what they share one of the chains for the receptor. Certainly IL-13 also has different signaling pathways that can explain those different mechanisms, right? But it's interesting in the case of 4 and 13, the signaling pathways that you're mentioning.
Yeah.
I had another question too.
Yep.
Hi Juan, I'm Tanu.
Hi, Tanu.
I wanted to ask whether the production of mucus is just a consequence of the infection and inflammation, or is it helping in the tissue repair too?
Right. So what it's known is that the mucus is induced, or it's poised to promote the worm expulsion, right? So these parasites, let me actually go back here. You see, for example, in the case of hookworms, which are the ones that we're going to be focused on, they have these teeth-like structures, right? Mm-hmm. They use those to really hang into the tissue. Other parasites use other mechanisms, right? Roundworms, certainly, they dwell in the tissue and they stay there. So the idea, or what is proposed in the field, is that this mucus production makes the surface kind of slippery and really tries to remove the worm. If it contributes to the wound healing, I'm not aware of that, but I would not be surprised if it does, right? Especially in these mucosal sites where you need to have that. I mean, the importance of that mucus layer is it's very well established when we talk about microbiota and how the regulation of that needs to be in terms of the epithelium. Again, to the best of my knowledge, I don't recall the mucus directly participating in the wound healing, but I would not be surprised if that is the case.
Okay, thank you.
Yep. All right. So speaking more about these basophils, right? Why were we interested in the basophils in the first place? As I mentioned before, this is a rare population of immune cells. For many years, actually, people doubted, or the field doubted, if this population of cells even existed in mice because they were hard to find, but they are there. Even though they are small in numbers, they certainly expand in the context of Helminthic infection, but also allergic diseases. Perhaps one may argue that basophils are mostly known to participate in the context of allergies. In the context of helminths, they are also activated by a number of pathways that are known to participate in the context of a helminth infection. That includes cytokines such as IL-3 and IL-33, as well as the cross-linking of the IgE antibodies that I mentioned that they are produced in the context of a helminth infection. So when these antibodies bind to the surface of the basophil, and then the antigen cross-links the IgE, this activates the basophil. And in response to these signals, they are known to secrete robust amounts of different effector mediators. And you can see some of those here, such as histamine, and they are mostly known to secrete histamine, but they certainly secrete other bioactive lipids, such as prostaglandins and leukotrienes. Especially, they are known to secrete a lot of IL-4 and IL-13 type 2 cytokines right there. So really, these are potent cells that secrete a lot of those. And for many years, again, these cells, I would argue, they were popular as targets to treat allergies, but really how these cells contribute to anti-helminth immunity is not very well understood. And it's not very well understood because one of the very first systems that was used to study these helminth infections was the use of animal models, right? And one of the most well-established animal models to study anti-helminth immunity is infections with a hookworm, Nipostrongylus brasiliensis, or for friends like mine, Nipo. And Nipo is the animal model for human hookworm infections. The hookworms, they have their name because they have that hook that I showed you in the beginning that they use to anchor themselves to the mucosal epithelium.
How small are these worms?
They are, you can see them in the microscope for sure, with the 5x objective. As they go through different larval stages, they grow, right? So the L3s, so I'm gonna go into what I mean into that in a second in the life cycle, but the L3s, you can see them in the scope. And even with your, with your blink, with your eye, you can see tiny little things. They look like tiny spaghettis. The adult worms, though, they are big. The adult worms in the intestine, they are, I would say, a like 2 centimeter, maybe like 1 centimeter long, maybe. But you can see them for sure. Yeah. Yeah. So again, these are big, parasites. They are not your usual bacteria or viruses. And the model with Nipo was not only well established to study type 2 cytokine responses, but also for pretty much for over 40 years now, it was recognized that the infection with Nipo of mice induces expansion of basophils, right? So really that implicated that basophils were doing something in the mice. And as I mentioned before, this model is quite useful to study type 2 cytokine responses because it also allowed to establish the role that many of these cells have, right? In this model, it's known that the early days post-infection, the cytokine responses are dependent on the activity of these innate immune cells, in particular the ILC2s. But later, the cytokine response is also taken by the Th2s and also the M2 macrophages in the late stages too. So really, this was one of the most well-established models to understand this type of cytokine response. So before, another thing I want to mention is the life cycle of these parasites. We're gonna use some aspects of it, and these parasites, after they are inoculated in the skin of the mice, usually in the wild, hookworms, the way they infect humans is they penetrate the skin of the people walking, without any shoes or protection in the rural areas. So hookworms are able to penetrate the skin. We mimic that by inoculating the larvae into the skin of the mice. They use the blood circulation, and these parasites, they move fast, I can tell you that. And they use the circulation to burrow into the long parenchyma within 24 hours post-infection. They have to do that in order to continue with their development, and it's not really clear why they have to do that. And then after 2 days, they migrate out of the lungs, and they pretty much they are involved in a process that is called cough and swallow, right? So the larvae are cough out of the pulmonary system and then swallow through the esophagus. I know it sounds gross. I really hope everybody already had breakfast.
I haven't yet, but it's fine. I won't have it today.
Yeah. No, this is what it is, right? So the larvae are cough and swallow by, in the case of human hookworms, by patients, or in the mice by mice with Nipo, and then they reach the intestinal tract, where the adults by day 4 they start mating and producing tons of eggs that are then secreted in the feces. You can collect those eggs in the feces and culture them to have an experimental system. But the reason why I have this here is because the model with Nipo is quite useful to understand the type of cytokine response, because as I mentioned, one of the functions of this response is to promote the worm expulsion, right? And mice have a natural ability to expel the worms approximately by day 11. And this natural ability to expel the worms is heavily dependent on that type of cytokine response. So with that in mind, we proceeded to ask, do basophils contribute to this expulsion of the worms right here? So for that, we use a well-established model of basophil depletion. I'm gonna go into
Sorry, I
that.
have one question.
Yeah.
Yeah, I wanted to ask you because a few months ago I read that some parasites can hide from immune system, for example, in adipose tissue. Is it the case with this Nipah worm?
Yeah, right. That's a good question. That is not the case for Nipah, because it's the life cycle of this parasite really involves the lungs and the GI tract, to the best of my knowledge. I think the one you are referring to is Trichinella. Trichinella spiralis, which I also work with that. It's a little bit different, the life cycle, but what those parasites do, and it's really cool, is that after they reproduce in the gastrointestinal tract, the newborn larvae, the babies, they go into all the sides of the body. They really have the capacity to go into the muscles, into the fat, right? Trichinella, it's mostly disseminated in the population in humans by eating, uncooked meat like pork or stuff like that. And you find those larvae in the form of cysts in the muscles, but that is not the case here with Nipah.
Okay, thank you.
Yeah. Okay, so we took our basophil depleted and just asked, do they have more worms? And importantly, where we depleted the basophils, we didn't see much changes in the worm burden of the mice compared to control, as you can see here. And even though this data was disappointing, it was definitely not surprising because different other people have seen the same phenotype. So this was what I meant with why are basophils or basophil populations expanding in the context of an Nipah infection? When you deplete them, you don't see any changes in the worm burden, right? So this really posed a question for us, and an opportunity to ask, if they don't really contribute to the worm expulsion, do they participate in other aspects of this type of cytokine response, right? So we hypothesized that they might contribute to the wound healing. And the cool thing about the Nipah model is that it also allows you to study that aspect. And the way you do that is because during the life cycle of the pathogen, right, when the larvae burrow into the lung parenchyma, as you can see here, the parasites, and perhaps you can appreciate more the damage that these parasites cause by this picture right here on the right, where you can see the larvae literally dwelling in the pulmonary parenchyma and inducing robust amount of hemorrhage and tissue damage. You can see the hemorrhage or the extent of that by the gross pathology on these pictures right here. But what's impressive, in my opinion, is that the type of cytokine response, once the parasites are gone, right, because they only stay in the lungs for 2 days and then they leave, the type of cytokine response is now in charge of promoting the wound healing of the lungs. And the cytokine responses are actually quite efficient at controlling the hemorrhage and promoting the wound healing of that. So then we asked whether basophils participated in this process. So the first thing that we did, and now I'm referring more to the paper, was to address whether basophils infiltrate the tissues, right? Perhaps I missed to mention that basophils are mostly circulating in the blood. They are in the blood. So, however, we wanted to know whether they even infiltrated the pulmonary parenchyma. So for that, we took naive mice or mice infected with Nipah, and at 3 days post-infection, we injected the basophil-specific antibody for flow cytometry, anti-CD200R, in the bloodstream of these mice.
What's the function of this protein, the 200R?
It's in the basophils, I don't believe it's super well understood. In other cells such as macrophages or alveolar macrophages, it's supposed to promote actually regulation of these cells. Okay. But I don't recall this protein being super well defined in basophils.
So would this anti-CD200R also deplete some macrophages?
Well, it's not depleting them in this system because we only inject them 5 minutes before the harvest.
Oh.
So this is not gonna. So with this strategy, we're not gonna deplete the basophils, right? So with this experiment, we're just trying to label them.
Okay,
and actually, I was gonna go that. So the question we're trying to address is: are these cells moving or leaving the blood circulation and getting into the tissues? So by injecting this antibody 5 minutes before, what is reported is that you stain the cells that are in the blood circulation, but you can't stain those that left already the periphery because the antibody requires longer time incubations to reach those tissues. So what we did is we took the cells from the lung tissue and we analyzed the basophil population by flow cytometry. We gated on pretty well-defined markers for basophil, CD49B and IgE. And then in those basophils, we asked, could they be stained by the antibody, right? And in the naive mice here on the left, you can see that the majority of the naive of the basophils in naive mice, they stain positive for the antibody, just indicating that they are in the periphery, right? Which is what we expect. Again, basophils are mostly blood-derived. But look at what happened post-infection. Portion of these basophils could not be stained by the antibody, right? And this suggests that these portion of cells of basophils have already left the tissues and they are infiltrating the lungs. And even though 8% might not be super shocking to you or me, day 3, it's actually interesting that day 5 we found the peak of the lung infiltrating basophils, and then they went a little lower at day 7. So this suggested that basophils infiltrated tissue post Nipah infection. I don't know if I know this technique is a little complicated, but please let me know if there is a question.
So is the point of IgE just that there will be Fc epsilon receptors and they will be grabbing IgEs and that's what you're tagging? Okay.
Yeah. So you can either stain for the Fc epsilon receptor or stain for the IgE that is going to be bound to the surface of the basophil. All right. So, but when we look closer at this kinetic response, we actually can get an insight why basophils might not actually participate in the worm expulsion, right? Because if basophils start infiltrating the tissue by day 3, and they certainly peak by day 5, by that time, the worms have already left the tissue, right? They only stay there for 2-day stops. So this really suggested that the reason why basophils might not be directly involved to the worm clearance is because they only enter tissue once the parasite has already left or has already been cleared from that. So this really suggested that basophils might participate in the wound healing process that occurs once the parasite is cleared. So
Juan, so I have a question. Do you look after day 7? So if you do like the kinetics more days, you will reach out to a 0 level? And if
no, we didn't. I'm afraid we only focused on the first days of infection, right? We only did it like till day 7. And
is this the same process happens also in the intestine? So if you don't have the parasite,
right,
then you will the basophils will arrive after that, or just a phenomenon that only happens in the lungs?
Right, that's a really good point. We didn't address whether this infiltration also occurs in the intestine. We didn't do that. That's a good point. Whether this process also occurs in the intestines, we don't know.
These mice die at the end of the infection, or are they just sick?
No. That's cool, right? So you're bringing a great point. So I just show you the hemorrhage. I just show you the larvae crawling from the trachea to the esophagus. The mice don't die. And yeah, they don't fare very well during that 48-hour post-infection period. You can see them with some difficulty breathing. After that, though, the mice are fine. And people actually, one of the aspects of helminths is that they can infect the people for quite a number of years, and they don't really have like big symptoms. They have malnutrition, they have growth impairment, they have anemia, but they don't die. And that goes back again to my point that helminths, as many other parasites, they require the host for their own survival, right? So it's not the type of infection that is going to kill you. It's the type of infection that is going to cause you morbidity because the parasite needs you to produce those eggs and then secrete them as long as they can. In the case of the mice, I should mention, Nipah, it's a rat pathogen, right? So it has been adapted to the mice. That is why the mice have this natural ability to expel them. But in rats, the Nipah infection induces a long, chronic infection. In mice, they don't do that. So then we address whether the depletion of basophils altered the capacity of the mice to breathe. And we did that by using a well-established model of basophil depletion, in which basophils, in which the mice carry the human diphtheria toxin receptors. Mice are naturally resistant to diphtheria toxin, but they carry the human version of the receptor under the control of the basophil-specific gene, MCPT8. And this is a previous strategy that has been used to only target basophils. It doesn't target other related cells such as mast cells. So following administration of DT into control and basophil depleted mice, we evaluated the oxygen saturation in the blood by pulse oximetry. And what we found here is that in the black and green lines, you don't see much differences when basophils were depleted in naive mice. But importantly, when basophils were depleted post-infection, in the red line here, you can see that that was associated with reduced oxygen levels in the circulation, as compared to control mice that were infected in the blue line. So this indicated that depletion of basophils is reducing the pulmonary function of the mice. So we then wanted to look at the tissue, at the lungs, at the pathology, just to see if it was different. And what we found is, again, when basophils were depleted in naive mice, we didn't see much changes in the pathology of the tissues, as you can see here. Oh, I think I'm going a little slow. I'm gonna speed up a little bit, maybe. But importantly, what we found is that when basophils were depleted in Nipah-infected mice, this resulted in the accumulation of cells in the tissues by H&E staining. And when we analyzed this cellular infiltrate by flow cytometry, one of the cells that changed or that was elevated was the eosinophils, right? As you can see here by flow cytometry, when we quantify the number of eosinophils as compared to control mice, they were elevated following the depletion of basophils. We also found that the airways of these mice, they were a little congested. So we analyzed the production of mucus. And we did that by two approaches, by PIS staining. And again, you can see here that depletion of basophils naive mice didn't really change the production of mucus. But importantly, when these cells were depleted in Nipah-infected mice, this resulted in the accumulation of mucus. We also evaluated the expression of the MUC5 marker, which is associated with that goblet cell activation, and the goblet cells are the ones that produce this mucus. And again, the mice that were depleted of basophils, they had increased expression of the MUC5 marker. So this really suggested that depleting basophils results in accumulation of eosinophils, as well as more production of mucus. And again, these effects are known to be driven by type 2 cytokines. So we look at the cytokine response in the tissues. So for that, we took biopsies of the lungs and analyzed the expression of type 2 cytokines by real-time PCR. And what we found was that the depletion of basophils resulted in elevated expression of IL-4, IL-5, and IL-13, as you can see in these diagrams, as compared to control mice. So this suggested again that the increased eosinophilia and the increased mixed production it might be due to elevated production of type 2 cytokines, specifically IL-5 and IL-13, that, as I mentioned before, they promote the eosinophil development and the mucus production. So then when we look back at the kinetic of this basophil response, right, we notice that the basophils peak around day 5. And by that time, in the NIPO model, it's well established that the inflammation is driven by ILC2 cells. So we hypothesized, or we questioned whether ILC2s were, the function of these cells was altered in the absence of basophils. And we were also interested in ILC2s because previous reports have shown that basophils can regulate the activity of these cells in the context of a lung allergy and also in the skin. So these ILC2s, what are they? So they are innate lymphoid cells that they have been shown to respond to a number of signaling pathways. I know this diagram is complicated, but we're not going to cover all of it. The point I'm trying to make, though, is that ILC2s can respond to a number of different signals, including cytokines, neuropeptides, and even bioactive lipids or vitamins. And in response to those signals, ILC2s are recognized to produce a lot of type 2 cytokines, including IL-5 and IL-13, as well as other effector mediators such as amphiregulin. And I already mentioned IL-5 involvement with eosinophil or IL-13 with the mucus, but also amphiregulin promotes the tissue repair. So we asked whether the activity of ILC2s was elevated in the absence of basophils. And to do that, we went back to the basophil depleted model. We collected the lung cells, and we re-stimulated to stim for cytokine production by ICS. And what we found was that the flow cytometry analysis revealed that the depletion of basophils resulted in increased percentages of IL-5 and IL-13 producing ILC2s, as well as increased numbers of those cytokine producing ILCs. So
this your eureka moment?
Huh?
This is like, I found something very cool here. Like, this seems like a great link between basophil depletion and ILC3s, right? ILC2s. If I had this experiment, I would be partying that night.
Yeah, that was cool, right? So we found, yes, so actually, yeah, I didn't emphasize that. I was a little worried about the time. Yes, so I have to be honest with you. When we started this project, the idea was that basophils secrete types of cytokines. If you deplete them, you're gonna have a reduced cytokine response.
Yeah,
I mean,
that's what you would
that
expect.
was the knowledge. Exactly. And right, like you said it took me a while, I have to mention, to believe that. I repeated it and repeated it and repeated it. Mark was also hesitant at the moment, but he was quite excited, like you said. I guess as Chris Hunter once mentioned in one of his talks, I guess that one of the dangers of doing hypothesis-driven science is that sometimes you so want to believe that your hypothesis is right, and when the data proves you wrong, follow the data. The data is the data. Yes, so I share your enthusiasm for this data. Was definitely not what we expected. Yeah. So IL-2 responses seem to be increased. So we asked whether these phenotypes recapitulate in the absence of T cells, right? I mentioned that T cells also participate in the production of cytokines. So to address that, we took RAC2 deficient mice that lack adaptive lymphocytes, right? Both B and T cells. And in this case, we depleted the basophils by using the basophil depleting antibody anti-CD200R3. It's a different antibody that I showed you before, but in the treatment, we treated the mice for every 2 days during the infection. And at day 7, we evaluated the same parameters that I showed you before. And this method to deplete basophils has been also used before in the literature. And importantly, consistent with the data in the wild-type mice, the depletion of basophils was associated with increased numbers of cytokine producing ILC2s, as well as eosinophils and increased mucus production, suggesting that this phenotype can occur in the absence of T cells. And at that point, we really focus on the ILC2. So let me just make a pause right here and try to tell you what, or summarize what I've shown you so far. Is that following the infection with Helminths, this result, the tissue damage, results in the activation of ILC2s, and basophils have been proposed to contribute to that activation. And this induces a number of effector mechanisms that promote the worm clearance and the wound healing. But what our data was showing at that point was that once the parasite was cleared, the basophils might participate in the regulation of ILC2 responses, which is required to put a brake on this system and promote the tissue integrity before the production of cytokines is just too much. So then at that point, we wanted to understand how were basophils regulating the ILC2s. But this also offers a nice opportunity to understand or to study how ILC2s are regulated. Because as you see in this diagram, that's why I used it. We know a lot about how ILC2s are activated, and this list keeps growing by the day. But the mediators that inhibit or limit the activity of ILC2s is not very well understood. So we really had an opportunity to also study how ILC2s are properly regulated. So to do that, we use two different approaches. We sort purified ILC2s from control or basophil depleted mice, and we did single-cell RNA-seq and bulk RNA-seq. I'm going to focus on the single-cell RNA-seq first. And importantly, we didn't see major. We were able to find 6 clusters of ILCs within the ILC2 population. We were happy to find that some of those clusters were identified by genes that are known to be expressed by ILC2, such as IL-13 and arginase 1, and others also have a REC. But importantly, the depletion of basophils didn't really alter the cellular composition. These clusters, they were not really changed following basophil depletion. What we found also was that the depletion of basophils resulted in elevated expression of IL-5 and IL-13 genes, as well as anti-regulin. However, these changes in the expression, they were not referred to specific clusters. They really, indicated more to the global ILC2 population. So then we went back. So one of the limitations of the single-cell RNA-seq approach is that the depth of the gene list that you generate might prevent their use to pathway analysis. So we use bulk RNA-seq to ask what pathways were altered in the basophils, in the ILC2s in the absence of basophils. And what we found was a number of genes that were differentially downregulated in the basophil depleted ILC2s as compared to the control ILC2s. And when we took those genes that were differentially expressed and performed pathway analysis, one of the top candidate pathways was the neuroactive ligand receptor interaction, the G protein coupled receptor pathway. So we were quite interested in this pathway because at the time, a number of papers have started to indicate that neuropeptides regulate the activity of ILC2 responses. And some of those neuropeptides that were highlighted included neuromedin U, BIP, and CGRP. These also extended to other neuron mediators such as neurotransmitters or even hormones. But we were particularly interested by the studies focusing on NMU because a number of studies have highlighted that neuromedin U, which also signals through the same pathway that we got by the pathway analysis on RNA-seq, and neuromedin U activates ILC2s. So we were quite interested in this family of peptides. Before I show you more data, just to touch base on these peptides, they are classified into 4 classes, specifically bombesin, kinesin, neurotensin, and neuromedin U. They are expressed in the CNS, but also in mucosal sites, such as the lungs and GI, and they signal through G-coupled receptors. These are short peptides. In the case of neuromedin B, it's a decapeptide right here. And as I mentioned before, after they bind to this to the G coupled protein receptors, they signal through different pathways. And they have been implicated in a number of functions, including some immunomodulatory properties. But really, the functions of these peptides in the context of a type 2 immunity is not very well understood. So we wanted to see how was the expression of the receptors for these peptides in the ILCs. So the first thing that we did was just to validate some of the data we got from the RNA-seq studies by purifying ILC2s from control and basophil depleted mice and evaluating the expression of the receptors by real-time PCR. And what we found is that ILC2s from basophil depleted mice, they had reduced expression of neuromedin B receptor as compared to those purified from control mice. And importantly, we didn't see differences in other neuromedin receptors, such as neuromedin U receptor. And also, when we evaluated the expression of the ligands, right, neuromedin B or neuromedin U, they didn't seem to be altered by the depletion of basophils. So at that point, we really focused on this finding that the ILC2s expressed fewer levels of neuromedin B receptor when the basophils were depleted. And we wanted to confirm that at the protein level. So we monitored the expression of neuromedin B receptor in ILC2s by flow cytometry. And as you can see here, while the ILC2s from naive mice, they express very low levels of the receptor, the post-infection, those levels are induced. However, when basophils are depleted, the ILC2s express lower levels of neuromedin B receptor. And we were certainly able to identify neuromedin B receptor in other immune cells in the lung, such as ABMAX, but this expression was not altered by the depletion of basophils. So this suggested that basophils specifically regulate neuromedin B receptor on ILC2s. So then we asked, can basophils directly promote neuromedin B receptor on ILC2s? So to do that, we purified both cells and then co-cultured them for 24 hours in IL2 and IL7, and then evaluated the expression of neuromedin B receptor. What's very interesting is that the co-culture of ILC2s with basophils resulted in elevated expression of neuromedin B receptor on ILC2s as compared to the ILC2s culture alone.
Do basophils require IL2 and IL7?
Yeah, actually, well, I actually made a typo here. We did IL2, IL7, and IL3. You require IL2 and IL7 to keep your ILC2s alive and happy, and basophils require IL3. So yeah, I forgot to put that one here, but we did IL3 too. But that's just to keep them alive. Yeah. So then we analyzed whether other mediators that are secreted by basophils can promote neuromedin B receptors. We didn't see that in the case of IL4, histamine, or PGD2. But importantly, when the cells were treated with PGE2, they expressed more or increased levels of neuromedin B receptor compared to the ILC2s alone. But this increase was not as high as the basophils. So this suggested that PGE2 may be one pathway by which basophils promote neuromedin B receptor.
In the last slide, I was wondering, I was looking at your experimental setup. So the basophils come from an animal that was just previously given by IL3. And where did you took those basophils, from the lung?
No, we took them from the spleen. I mean, so, we tried them from the lung, but it was too hard.
So this is basically saying that no matter where the tissue you are, basophils will promote NMB receptors on ILC2s.
Correct. Yeah, we didn't split them up or got them from the lung. That's certainly something to do. It's challenging because you don't get a lot of them, but yeah, this is just a proof of concept that basophils can do it directly.
Yeah, I mean, I was like wondering, what didn't you took the basophils from a Nipah infected animal?
Because
Or
you don't get enough of them.
What
yeah,
if you
we
give
try.
IL-3 too?
Sorry?
If you also give IL-3 to expand the basophils and plus Nipah infection.
Yeah, so in the model of Nipah, it's well established that the expansion of basophils is dependent on IL-3. And those studies are, you know, they have been well established. We tried getting them from Nipah infected mice, but you just don't get enough numbers. I mean, yeah, we try. So, I mean, that was the reason why. So, all right.
Okay, thank you.
Right. So what I've shown you so far is that basophils seem to they seem to regulate the activity of ILC2s by promoting the expression of neuromedin B receptor. So the next question we ask is, can this pathway actually inhibit ILC2s, right? And importantly, as I mentioned before, a neuromedin B receptor was also present in other immune cells. So we wanted to understand this pathway in the context of hematopoietic cells. To do that, we generated an NMB receptor flux mice in which the exon 2 is flanked by LOX-P cassettes, and we crossed this with BAV1 Cre mice that have been reported to target the hematopoietic compartment. So the first thing that we did, just to validate the system, we took the NMB receptor flux and the cross ones and evaluated the expression of neuromedin B receptor in different cell compartments. And as you can see here, in the flux mice, we didn't see changes on neuromedin B receptor in the CD45 negative cells, but we were glad to see reductions in CD45 positive as well as ILC2s in the flux mice that were crossed with the BAV1 Cres. And then we evaluated the expression of type 2 cytokines in the lungs. And what we found is that the targeting of this receptor in hematopoietic cells resulted in elevated expression of IL-4, IL-5, and IL-13. And I guess at this point, it's not that surprising anymore. This was associated with increased numbers of cytokine producing ILC2s, as well as increased numbers of eosinophils and mucus production. But importantly, targeting the receptor on hematopoietic cells recapitulated the phenotype observed in the absence of basophils. So this strongly suggests that one pathway by which basophils might operate is by promoting the expression of a neuromedin B receptor on ILC2. So then we use a gain of function approach by treating the mice with a ligand, neuromedin B, and see if neuromedin B is sufficient to inhibit Nipah-induced ILC2 responses. We use RAC2 deficient mice because, again, in these mice, since they lack adaptive lymphocytes, the inflammation is very dependent on ILC2 activity. And nicely, we saw that the treatment with neuromedin B resulted in reduced numbers of cytokine producing ILC2s, reduced numbers of eosinophils, and reduced mucus production. So this was consistent with all the readouts that we got before. What was interesting, though, was the pathology of these mice, because you can see here again that the treatment with the neuropeptide on naive mice didn't really alter the pathology. But when the mice were infected with Nipah and treated with neuromedin B, this resulted in this massive accumulation of immune cells in the lungs as compared to mice treated with vehicle. And importantly, when we look at the VAL, the appearance had more hemorrhage or more RBCs, and we also quantified those. So remember that I mentioned that the function of the type 2 cytokine response is to control that hemorrhage, right? So these data indicated that by inhibiting the type 2 response, NMB is also inhibiting the wound healing. So this was a proof of concept that neuromedin B can inhibit type 2 cytokine responses. And I really want to mention that previous studies by a number of labs, including Bill Gosses and Judy Allen, have demonstrated, and Nicola Harris have demonstrated that in the model, in the NIPO model, before the establishment of the type 2 cytokine response, there's a strong type 17 response that is dependent on neutrophils and IL-17. And when you block the Th2, you get an increase on these type 17 responses. So that was exactly what we found that the treatment with neuromedin B associated with increased IL-17 expression, as well as neutrophils, in the BAL and lungs, right? So those cells that I showed you before, they seem to be mostly neutrophils. So this indicated that neuromedin B, in fact, can inhibit ILC2. The last question we had is how is neuromedin B regulating ILC2 function? How is it doing? So that was the last part that we did. So to do that, we flow purified ILC2s from wild-type mice, and we treated them in the same conditions for 24 hours with neuromedin B. And what we found is that the treatment with neuromedin B was sufficient to inhibit the secretion of cytokines in the supernatant. But what was interesting was when you take the same cells from a basophil depleted mice, the ILC2 can no longer respond to the neuromedin B treatment, and is associated with the reduced expression of the receptor. So this is really suggesting that the absence of basophils is preventing the ability of ILC2s to respond to these neuromedin B in the tissue. So then we asked whether NMB alters ILC2 proliferation. To do that, we purified ILC2s from NIPO-infected mice, and we labeled them with CTV following standard methods, and we cultured them for 4 days in the presence of cytokines as well as in the presence of neuromedin B. We use PGE2 as a positive control to inhibit proliferation because it has been shown to inhibit proliferation. And what we found was that the treatment with neuromedin B didn't really prevent the capacity of ILC2s to proliferate as compared to control mice. And you can see here that the PGE2, as reported, definitely prevented the activity, the proliferation of ILC2s. And in green, neuromedin B didn't really alter that capacity of PGE2. Right. So this was actually interesting because we have here a pathway that seems to be targeting the secretion of cytokines by ILC2s, but it doesn't target all the functions, right, of ILC2s. And another finding that was interesting is similar to the 24-hour cultures that I showed you before, the treatment for 4 days with PGE2 is sufficient to promote the expression of neuromedin B receptor on ILC2s. So this really suggested that PGE2 and neuromedin B can synergize to target ILC2s. On one hand, PGE2 blocks cell proliferation. On the other hand, neuromedin B targets cytokine production. So that was exactly what we tested next by treating the cells with both neuromedin B and PGE2. And what we found is that when you treat ILC2s with both PGE2 and NMB, this results in a strong inhibition of the secretion of cytokines. So this is really suggesting this cooperation between these two signals. But again, we were quite interested on how is it possible that NMB targets exclusively cytokine secretion without altering other functions of ILC2s. So the last experiment we did was just to take the ILC2s, treat them with neuromedin B, and then analyze the transcription profile by RNA-seq. And we certainly didn't see many changes differentially expressed when we treat them with neuromedin B. But one of the top genes that were downregulated by NMB treatment was a P2RX7 gene. And this gene codes for the P2RX7 receptor, which is a sensor for extracellular ATP. And this receptor has been shown to respond to extracellular ATP, and it's involved in a number of processes, including the activation of the inflammasome and the survival of T cells. And it can be inhibited by pharmacological strategies such as the use of BBG. And we were interested in this gene because just 2 years ago, it was published that treatment with BBG induces the suppression of ILC2 numbers as well as expression of cytokines in a different model of helminth infection. This was with H. polygyrus. So we then wanted to see if we just target P2RX7, can we mimic the effects of neuromedin B? And so we did exactly those in vitro studies, but instead of treating them with neuromedin B, we treated them with the inhibitor for P2RX7 receptor. And we found that the treatment with the inhibitor recapitulated the effects of neuromedin B on the production of cytokines by ILC2. So that was my last data slide. So really to put all of this into a model, what this suggests is that, again, after you have this tissue damage that induces the activation of ILC2s, which is required to promote the worm clearance, right? In order to regulate the activity of ILC2s, basophils promote the expression of neuromedin B receptor, allowing these ILC2s to respond to these neuron-derived mechanisms such as neuromedin B that inhibit ILC2 function, specifically very strongly in their ability to secrete cytokines. And all of this is required to promote the integrity of the tissues. And with that, I just want to thank my research advisor, Mark Siracusa. He has remained a mentor to me even after I left his lab. Members of the Siracusa lab, including John, Christina, Hannah, and Jenya, other former members of the lab, the collaborators that they still have at Rutgers, as well as the facilities and the funding, and of course, you guys for the invitation to present my data here. And I'll be happy to take questions.
Just before we start, anybody in the audience can also ask questions in the chat, and we will we'll relay the questions to Juan. That was superb work. I would have partied 4 or 5 times if this was my publication. Like after every figure, I realized something. I'm at the Taco Bell because I'm poor, so I go to Taco Bell.
I can tell you that there were parties. There were definitely parties. One of the nice experiences I had with Mark is that whenever we had this huge harvest, he likes to buy us pizza or just give us money for food, like just buy you guys some dinner because you're gonna be here late. So, if you guys are ever interested in helminth immunity, Mark is one of the best mentors, in my opinion, to do that.
After this, I think I am a little interested. I like this research where you have multiple cell types that you would think are completely unrelated, but they come together and form this, they complete this puzzle. Like I wouldn't expect neurons to be doing anything when it comes to inhibition of inflammation.
Right. Yeah. So the whole field of neuroimmunology, it's really booming. I mean, I think we just realized that at mucosal sites, and this is one of the reasons why I'm really interested in mucosal immunology, right? You have these communication events between neurons, epithelial cells, tissue resident immune cells, right? And see the interactions between these neurons. We have one mechanism here, but I'm certain there are many others, right? I mentioned the neuromedin U aspect, so you can argue that neuromedin U, the work from David Ortiz, Enrique Vega Fernandez, and also Vijay Kukro, they really demonstrate that neuromedin U is a strong activator of ILC2s. On our hands, we are proposing that neuromedin B does exactly the opposite. So this almost looks like a rheostat between neuromedin U and neuromedin B.
Which program did you use to draw this figure? It's pretty cool.
Oh, it's a fancy program. It's called PowerPoint.
Really? you made this in PowerPoint?
So Mark, he did the cells, right? He did the neuron, he did basophils, the goblet, the eos. He shared them with me, the figures. So I think he used what's it called? Forgot the one that's online.
BioRender?
No, BioRender. I think so.
Okay.
Yeah.
Yeah, the figures look pretty cool.
I certainly give total credit to those to Mark. He was nice enough to share them with me. Yeah.
It's pretty good stuff. Do you guys have any questions?
No.
Oh man, I'm still just trying to absorb. There's so much stuff in this paper, and the final point when it all makes sense. It's so fulfilling.
Yeah, no, I apologize if I went fast. And I look at the clock and I was like, oh my God.
Oh no, not at all, not at all.
I tend to
No,
talk.
it was very informative.
I think the slides were very clear about what you want. Like you have a question on each slide and the answers. It can't be clearer than that. So yeah, I love the present. I love this paper. When I was reading it, I thought to myself, well, these 10 figures are worth it.
No, thank you. No, I really appreciate those comments. It was definitely a lot of work. Many of these things didn't work, and many other things changed because of the pandemic, right? I had some experiments in line, back then in March, and then we went to lockdown and many things. But I'm glad this is out. I'm glad you guys like it. We really are trying to put out the concept there that well, there are a number of take-home messages, I would argue. Number one, basophils are not the bad guys that only participate in allergies. They have some good functions, right? Number two, they don't only activate, right? I mean, here they seem to really regulate. So I know it's, for example, like Eugenio's questions, right? It's soil purifying or studying basophils is challenging for sure, especially in the tissues. They are so few, but there is value on that, right? So hopefully with these RNA-seq platforms that you only need 10,000 cells, right? Yeah. This can really allow us to study this rare population of cells. And number three, in the tissues, in the mucosal sites, you just mentioned it, Justin, that these cells, neurons, epithelial cells, immune cells, both the tissue resident and also the ones that infiltrate, they engage into these communication pathways, and that's really where the action is. I would argue, right? That's really when a cell needs to decide whether, I mean, of course, it doesn't have consciousness, but it needs to respond to mechanisms that dictate whether it's producing cytokines or effector mediators or expressing surface molecules or not. And understanding that balance, understanding that reset has a lot of value, not only therapeutically, I would argue, but also biologically, right? To maintain that balance.
Yeah. And as you mentioned in your discussion as well, that this paper goes very well with this concept that as soon as the immune system activates itself, it puts in these checkpoints to bring it back down. And you see this even in antiviral responses, that as soon as you have interferon signaling, multiple inhibitors of IRFs, Janus kinases, they get activated. So if you prime a cell with interferon and you give it another interferon in next day, the cell will not be making as many antiviral proteins because it's got used to it and it's now knows that it's not supposed to be inflamed that much. So it's just a pretty I would say sophisticated system that's everywhere. Type 1 immunity, type 2 immunity. It's just so cool to look at these inhibitory mechanisms put in place in every step.
Oh, sorry, I think I shared again. Sorry.
Oh yeah, no problem. Okay, so
something I was not supposed to. Yeah.
So if you guys don't have to add anything else, Eugenio, do you have anything?
Yes, thank you, Juan, for once again great presentation. So, so I was looking at some data from Christina Schwartz, actually, who from Germany, who showed that basophils have a protection role, and this is somehow the question I always do it, but like I'm quite sure that if basophils, as you show at the beginning of your slide, you show that basophils have like no any role for at least at day 7 for controlling the worm burden. But you know, like my question always like relies on if the NIPO model would be available to do like a model of reinfection and to see if in the context of reinfection, the basophils will actually have a role. And at least it seems that in my second question will be, would you expect to see the same phenotype, all that you have shown us in the H. polygyrus model, or just like particularly in NIPO?
So let me just make sure I get, so the first question is, are basophils doing this in the intestine, right?
Yes.
Especially in the context of a second reinfection. That question has a lot of implications that are really cool too. And I even talked to David Veringer, which is the senior author, and Chris too. Remember, that was last year. So in the NIPO model, the reinfection really occurs. The protection against reinfection occurs at the level of the skin and the lungs. A lot of times the worms don't even make it to the intestine, right? So that's important too. And basophils, like Chris showed, and like David Veringer and Rich Lockley too, they show that post-infection in the reinfection, basophils are necessary for that protection, right? And they've shown that that again further contributes to my point that they must be doing something. The fact that these cells expand in numbers, they suggest that they have to be doing something. So if this replicates in the intestine in the second, well, first we'll have to go back to the intestine and see in the primary, right? Because once the parasites are cleared, there's also, tissue remodeling in the intestine. So we'll have to first address whether this occurs in the primary infection, right? That's something that, it's left to do. For about the second question with the model of H. polygyrus, just to introduce the model a little bit, it's a little different from NIPO because that worm is inoculated only orally by oral gamage, and really the life cycle of that pathogen is very restricted to the intestines, right? Doesn't have these migratory cycles. Secondly, H. polygyrus has or forms these granulomas in the intestine, and it really establishes a chronic infection. So if the mice are not treated, the parasite is not cleared, right? So I would argue that basophils might not do the same thing because the parasite is never cleared unless you do it pharmacologically, right? Now, I haven't done it, right? So the idea will be to actually do that. Take mice, hit them with HP, clear them, and then do your basophil depletion, right? So that's something that, it's certainly something that you could do. What's the name of Elia? Elia Tate Wagner at UWash. She have shown that in the model of trichuris basophils in the intestines, they promote type 2 cytokine responses, right? And that was with trichuris muris, like yet again another worm. So you're actually making a great point here. Most of the times we refer to these helminths as a group. That will be almost similar to as if I refer to Salmonella, Streptococcus, and or Enterobacter as just bacteria, right? But it's really important that we understand that helminths are very diverse. They have different life cycles, and not all the parasites are going to be the same. So one thing that I've always wanted to do is to take these cells, basophils, ILC2s from different helminth infected models and see what's different, right? I mean, I'm perfectly aware and I believe that you're going to see many things shared, but how different infections tailor or modulate the activity of your cells, name it, right? That would be actually really cool.
Thank you, Juan.
You guys, are there any other questions? Okay, I think this. Oh yeah, Maria.
Thank you so much for clarifying everything. I read the article and it was, for me, as not having much knowledge about basophils and ILC2s, it was a bit overwhelming, but your presentation clarified it. And I feel like I'm really more informed, and it's great because some immunology courses just, skip through ILCs and they only tell very short that basophils and allergies and things like that. So yeah, thank you very much.
No, I appreciate it. I'm glad you like it, Maria.
Yeah, the only thing they teach in Basophils is that they have an Fc epsilon receptor and they give you food allergy.
Exactly.
And
Yeah.
and they put them in the same basket as mast cells. I had no idea what's the difference between mast cells and
Right.
basophils.
Yeah, you just said it, right? Mast cells are super tissue resident. I show you that basophils are blood derived, are circulating. So that's one of them, right? They differ also in the expression of the proteins, right? That the strategy we use to target the basophils with MCPT8. So MCPT8, the name is mast cell protease, mast cell protease 8. So this family of proteases, they were firstly defined in mast cells. The MCPT1 is really involved in mast cell functions. I also had the opportunity to work on a mast cell project in Mark's lab. So we often target or use MCPT1 as a readout of mast cell function. MCPT8, in the contrary, doesn't really seem to target mast cells at all. It's also very basically specific, right? So you're right. You're right in the fact that there is value on studying these cells, even though they are rare. I actually wanted to, because I found that you guys are famous for your memes or GIFs, which I certainly enjoyed. So I wanted to share one with you of my own making. So this was back from my thesis defense, right?
In your thesis?
Yeah, in my thesis defense, when I defended my thesis, that was back in August. So look at this one. I think I may actually even found this from your website. But again, basophils are mostly known to promote anaphylaxis over peanut antigen, right? And they use this from SpongeBob. But there is this other show. I really recommend it. Cells at Work. It's in Netflix. It was in Netflix. But and they touch base on different immune cells. But when
Hold on, is it Cells at Work?
Cells at Work.
Oh, I'm yet to watch this. I've watched first 3 episodes. Anybody watching us right now, I'd 100% recommend watching Cells at Work. It's a good primer on immunology and it's an anime.
Yeah. I second that, absolutely. And it's cool that they do have an episode about Helminthic, and they speak about eosinophils, they speak about mast cells. When they touch base on basophils, it's really interesting. I'm not gonna give any spoilers, but they picture basophils as this, mysterious character that we don't really know much about, which is absolutely true, right? Again, it's a rare population, it's hard to study, but there's a lot of value on studying these cells. That was all I was trying to share.
I think this will be a good point to wrap up our discussion. Thanks a lot, Juan, for joining us today. And
thanks for the invite.
We would actually like, if you're welcome to join us as a panel member in future, if you'd like, whenever we have a type 2 immunity paper, we'd love to have you again. And everybody who's watching us, yeah?
No, yeah, that'd be great. Yeah, just, let me know. I think you guys do it every weekend, right?
It's twice a month.
It's twice a month. Okay.
Yeah.
Yeah.
And everybody in the audience, thanks for joining us, guys. We'll see you in 2 weeks. We have another guest speaker booked, and I will let you know on the Facebook page about the specifics. Thank you all for joining.